I have to. Okay, it's okay. Now, okay, we are streaming. Adilson, do you want
Yes.
Share the link? Okay, you are seeing? You can see?
Yes, I am seeing in my cell phone.
Okay.
It's transmitting now. I think we don't need to share because it's only a link from the transmission. But after I will send, Adeline will send the link to the in-hay team.
Yes, perfect.
Transmission. But yeah, good to know that it's working. It's better to try to test.
Yes, and the audio as well, because last time it was the audio that was not working.
Yeah, I didn't put the audio. Do you test it, Vanya, if it's working?
Oh no, it was not the audio. Sorry, it was the video.
I can share the link with you. Just a minute.
Yeah, I think after the presentation, the video will be saved, and so we can check if it's
of course
audio or not.
Yeah, good. We can do that.
Okay, perfect. So can I start?
Yes, we can do.
Yes. Okay. So hello again, everyone, and thank you for attending my rehearsal. And my project is called Resilient, Sustainable, and Nutrition, PGMP protein fractions as high potential food ingredients. And this work was developed in a joint program between São Paulo State University in Brazil and Nantes University in France. And during my stay in France, I was hosted by INHAE in the ESD team. And my supervisors are Vânia Regina Nicolette, Claire Berton Caraban, and Adeline Bois. So to start, I will give a little of context about the plant-based protein scenario. So according with the data of the FAO, around 95% of the world's food needs are provided for by just 30 species of plants. So this number is very small if we take into account that we have around 30,000 plant species that are edible in the world. In addition to that, if we think about plant-based proteins that have been used to develop new products for the market, we will see that soybean is the main global legume crop that has been used, and there is still a lack of pulse diversity in the products that we can find in the market. So this tells us that we need to diversify our sources. And when we talk about pulse crops, there is a group of pulses that are still considered underused in the world, or also called orphan crops. And most of these species are highly drug resistant, which its characteristic is very important in the context of climate changes that we are facing currently. But there are the production are almost all restricted to small-scale farmers in their native countries, and have been received very little attention of different segments of the current agriculture system, like researchers, private companies, farmers, extension service, policymakers, and decision makers as well. So PGMP is part of this group of pulses that are still underused. It belongs to the Fabaceae family, and the seed composition presents a protein content that can range from 18 to 28% in dry basis. So it can be considered a pulse very rich in protein. And the species is native of emerging countries in Africa and Asia. And today, India is both the largest consumer and producer of PGMP, accounting for 90% of global production. But the seeds were also introduced to the Central and South America by the slave route from Africa in the 17th century, and it adapted very well to the different climate conditions. But in countries like Brazil, the seeds are mainly used for animal feed. But this is started to change since some public institutions like Embrapa have shown interest in developing new cultivars that are more suitable for human nutrition. And when we think about world production from PGMP, according with the Global Economy of Pulse report from the FAO, PGMP is among the most important pulse in terms of global production and consuming quantities. But as you can see in the graph compared with the other pulse, the production is still very small, and it accounts for around 6% of total post-production across the work. And if you want to change the status of PGMP from an underused crop to a more used crop, we need to bring more scientific knowledge about its seeds and potential ingredients to generate more innovations in this section. So in the first part of my PhD, I did a literature review about PGMP works that have been published. And I saw that most of the works focused in the protein fractions is the type of ingredient that is most studied in the literature. And these works account for around 42% of the articles that have been published. And when we think about protein extraction methods that have been used, we can see that most of the papers focus in the conventional method that involves the alkaline extraction of proteins followed by isoelectric precipitation. And the other methods also involve some precipitation steps, and this can alter the conformation of proteins. So today there is an interest in study different methods of obtain protein extracts, possible with a way that is more muted to protein to maintain the structure of proteins in their more native state. And regarding functionality, around 16% of articles evaluate some functionality of protein fractions, but most of these articles focus only in a screen of different functional properties. And I found two papers that focus in a more in-depth study of a specific functionality. It was the case of these two papers of a group of Argentina that focused in the emulsifying and gelling properties of PGMP proteins. There is no work for foaming properties, for example. So in this thesis, I tried to evaluate different samples, starting by the conventional used precipitation-based method extraction to obtain protein concentrate. And after, I also proposed a muted extraction only with solubilization of proteins without any precipitation step. And the third method involved a muted extraction followed by purification of proteins by chromatograph techniques. And regarding the research axis of this thesis, we can divide it in three parts. So the first axis involves studies about the composition. And when I say composition, it's not only based in protein composition, but also lipid and carbohydrate fractions. The nutritional quality was assessed by investigation the effect of some seed pretreatment and also protein fractionation in reduce some anti-nutritional compounds like trypsin inhibitors, for example. And functionality was assessed by, at the beginning, I did a screen of different functional properties, and at the end, I focused in only one, that was the foaming property. So the objective of this work was to extract, purify, characterize, and evaluate the functional properties of different protein fractions from PGMP. And to do that, the work was divided in three parts. So the first part involved a study about the pretreatment effects on the composition and functionalities of PGMP seed ingredients. The second part was focused in the preparative scale purification of PGMP7S globulins. And the third and final part was focused in the air-water interface and foaming properties of crude and purified globulins. So in the first and second part, I used commercial seeds from a Unesp cultivar. And in the final part, I also used seeds from Pereira that was donated by Embrapa. So going to the findings, starting by the first part. So this work was already published recently, so I will focus only in some results that are more relevant to my presentation. So here, the idea was to investigate the effects of soaking and boiling pretreatments, not only in the digestibility and the reduction of anti-nutritional compounds that are good from the digestibility of the seeds, but also the effect in composition and functionality. So regarding PGMP, if we think about works that focus in seed pretreatment, we'll see that most of the papers evaluate only the effects on flour. And only one paper was focused in the effect of this pretreatment in protein fractions. And thinking about composition of PGMP ingredients, again, we will see that most of the works investigate the composition of flours, and only two papers focus in the composition of protein concentrate. And in previous work of our group, we saw that non-protein compounds in specific should be further characterized because they can account for a significant amount of the protein fractions. So with this, we saw a lack that regarding the effect of the functionality of protein concentrates and or isolates from pretreatment seeds. And also there is a lack of more in-depth studies of the effects on composition. So the main question that we wanted to answer here was how does seed pretreatment affect the composition and functionality of PGMP-based ingredients? The procedure that was used in this part of the study. So I had one control that was consisted of raw seeds, seeds without any pretreatment. The soaking was conducted against distilled water for 24 hours. And finally, soaking plus boiling, the boiling was conducted in soaked seeds for 30 minutes. And after the pretreatments, the seeds were dried and milled to produce the flour. And the flours were used to produce the protein concentrate using the precipitation-based method. Composition of the flours and also the protein concentrates. And the flours present a protein content around 20%, a carbohydrate content of 64%, and a lipid and ash content of 3 to 4%. And when we take a look in the protein concentrate composition, you'll see that we increase the protein content by threefold, and we decrease significantly the levels of carbohydrate, but they are still significant from 9 to 15% of the total composition of the protein concentrates. And we also observe an accumulation of lipids in the protein concentrates that was produced with raw seeds, and the levels of lipids decrease with the pretreatments that we apply. But in general, we also observe a significant amount of lipids that range from 4 to 13%. And the carbohydrate and lipid fractions were further characterized in terms of using liquid chromatographs, and we saw that they are present in different groups, as you can see, and some of them have impact in the nutritional quality and also in the functionality of these ingredients. So the main highlights of this part are that the carbohydrate and lipid fractions can alter the functionality of protein concentrates, and together they can account for around 30% of the composition of protein concentrates. Also, protein concentrates obtained from soaked boiled seeds show higher starch levels and absence of phospholipids, and these compounds can also affect the functionality of these ingredients. And the most important thing is that due to the complex composition that we observe from the protein concentrates, it's difficult to evaluate the contribution of proteins and non-protein compounds to the functional properties. So from this, we saw the need for a simpler system to understand and characterize the intrinsic properties of individual proteins, like the 7S globulin fraction. So this leads us to the second part of the research that was focused in the development of the preparative scale purification protocol for PGMP globulins. And so, as I said, since the protein structure is a mix of different compounds, we can find different protein groups, lipids, pigments, phenolic compounds, and other compounds. And since we want to study the intrinsic properties of a target protein, we need to first eliminate or reduce the interfering compounds that we have. So we can use liquid chromatography techniques to separate and purify our target protein. And regarding PGMP purification, some works in the past focused in the separation and purification of the 7S globulin from PGMP using analytical scale chromatographs. So this started by a group from India that published 3 papers about the separation of globulins from its seeds. And more recently, other groups also show some interest in investigate that. And the main findings of this work involve that PGMP globulin, 7S globulin, is a trimer of molecular weight of around 108 kilodaltons. These proteins were also identified as a glycoprotein and present a high homology with soybean 7S globulin. But we also identified some gaps in this work. So the first one was the own methods developed using analytical scale chromatography only. And the proteins were extracted using methods with precipitation steps. and the functionality of the purified proteins was not evaluated. So as far as we know, no procedure to preparative scale purification of PGP proteins has been developed, which will be of interest to obtain significant amount of samples to also probe functional properties of these proteins. And all this using a Mildred process to maintain the native state of the proteins as much as possible. So the main question that we wanted to answer here was how to separate PGMP7S globulin using a preparative scale purification process. So the process that I developed consists of a three-step liquid chromatograph protocol. And this process was first developed and optimized an analytical scale. And after we upscale the process. And in the first step, a mildly structured protein extract was injected in a desalting column. And in this step, the idea was to remove salt and other minor compounds that can form aggregates to improve the retention of proteins in the following steps. So here we injected the extract in the desalting column, and the proteins passed through the amorphous resin, and small molecules were retained in this resin. And at the end of this stage, we will obtain a desalted extract. So this desalted extract was injected in an anion exchange column that was packed with a positively charged resin. And in that way, we separated proteins based in their net charge. And at the end of this stage, I collect the 7S globulin fraction. So this fraction was further purified by injecting it in a size exclusion column with a resin with a wide range of molecular range. And here we separated proteins based on size. So to do this, I used two different approaches. The first one consisted of dialysis steps between this second and third chromatograph step, and after the gel filtration separation as well. And the dialysis was conducted for several cycles against ammonium carbonate buffer. And the second approach consisted in the direct injection of one color to the other without dialysis step. And here, the salts were removed from the fractions by injecting it directly in the gel filtration column, equilibrated with ammonium carbonate buffer. And in that way, we remove the salt the same way that we separate the proteins by size. So the bulk fractions that we obtain here were characterized in terms of proteomic analysis, SDS page, intrinsic fluorescence, trypsin inhibitor activity, and protein and carbohydrate composition. So starting by the first process with dialysis step. In the first step, I obtain a chromatogram with three different peaks, being the first one, the peak that is rich in proteins, as we can see in the SDS page results here. And the second and third peak consisted of minor compounds, possibly some phenolic compounds that were bound to the column. And as you can see here, we don't have much proteins in it. So I collected the first fraction and I injected it in the anion exchange column. And here again, I obtained three peaks, being the first one, the proteins that were not bound to the chromatographic column. And as you can see, it consisted of a mix of different protein bands. The second peak was the peak of 7S fraction. As you can see by the bands, they are characteristic of PGMP7S globulin. And the third peak presented the same two bands from 7S globulins, but very less intense. So I collected the second peak and I reinjected it in the size exclusion column. And I got one main peak, as you can see, and other small peaks that correspond to some aggregates that we removed. But here we observed the hydrolysis of the protein that we are purifying, as you can see by the difference in bands compared with the other SDS stage. So it was a result that we weren't expecting, and it shows some products around 25 kilodaltons, as you can see. So with this process, I obtain a partial hydrolyzed 7S globulin, and I will discuss further about these possible causes in the next slides. But first, I will present the other process. So using process 2 without dialysis steps, I saw the same two results from the desalting and anion exchange steps. But when I directly inject the extract in the size exclusion column, I was able to maintain the protein in its native state, as you can see here, comparing with the 2B unit of the other stage. So these bands were identified by proteomic analysis as the alpha chain and beta chain of the beta-conglycerine from PGMP. And that way, with this process, I obtained the native 7S-globulin. So the proteolysis that we observed with the first process, as I said, the reasons for that are unclear to us, but I put here some important things about it. So the products that we observed were two bands around 25 and 20 kilodalton that came from the alpha chain from PGMP7S-globulin according to proteomic analysis. And we observed a similar phenomenon in the literature where some authors observed that proteolysis of the alpha chain of soybean 7S-globulin, whereas the beta chain was resistant to hydrolysis, so it's the same case as ours. And according to the authors, this proteolysis can happen due to some proteases that are present in the flower and protein concentrate, even in mature seeds. So we try to identify the sections that exhibit some enzymatic activity in our samples, in the flowers and also in protein fractions. And we found this protease, the aspartic protease in the leucine aminopeptidase, that could be responsible from this hydrolysis. But given the alkaline pH conditions used in our extraction, that was pH 8, and also the fact that we add EDTA, it's not expected that this protease will find optimal conditions of action. So we exclude this hypothesis. About the cleavage position, Wilson and co-workers observed the cleavage of P7S-globulin around the Köppen domain, generating products from 19 to 28 kilodaltons. So it's in the same range of the products that we observed from PGMP, which means that probably the cleavage can also occur in around this domain. And the main highlight that we have from this part is that the hydrolysis that we observed in preparative scale was not observed in the analytical scale process, suggesting that aspects linked to the upscaling of the process, like the higher process time or higher concentration, may be related to optimal conditions for the action of this protease. Regarding the yield of the process, in these images we can see at the distribution of dry matter that is represented by the different size of arrows, and also the distribution of protein content during the process that is represented by the different intensity of color. And at the beginning of the process, I started with around 200 grams of initial flour with a protein content of 19%. So in that way, we got around 39 grams of total proteins. But according with the SEH-PLC in the naturation condition analysis, we observed that around 86% of total proteins are soluble in the PGMP flour, and that 48% of soluble proteins corresponds to 7S-globulin. So in that way, at the beginning of the process, we had around 16 grams of 7S-globulins that are extracted. And if you consider that at the end of the process, I got purified fractions with a protein content of 89%. And if you consider that all the mass that I got correspond to the 7S-globulin fraction, we can calculate the yield that range from 23 to 29% of 7S-globulins that were recovered. And this yield can be considered good according with data from literature when the recovery of 7S or 11S-globulins can range from 1 to 19% in lab scale purification, but there is no data from preparative scale purification process. So about the composition of the purified fractions, as I said, both fractions presented a protein content that was around 89%. And we also identify a carbohydrate content around 3%. We didn't quantify the lipid fraction because it demanded a lot of samples to do that. And we have only a few to do all the characterization and improve functional properties. But we expect that the lipid fraction will be low to this purified fraction. So about the protein composition by SDS stage, here we can see the typical bands that we got from the native 7S-globulin and from the partial hydrolyzed globulins. But the main thing here is that if we pay attention to the bands with small molecular weight, we will see that the first process with dialysis step seems to be more efficient in remove the contribution of other protein groups that we get in the samples compared with the one with injection in the gel exclusion, gel filtration column. And about the carbohydrate composition, we identified in both fractions a high mannose content, and that is a very interesting result, considering that even the fraction that was subject to extensive dialysis steps, we observed this high level as well. And this suggests that this mannose is strongly bound to proteins and can be related with a possible glycosylation of these proteins, since we know that globulins from PGMP are glycoproteins, and mannose can play an important role in this phenomenon as well. So I also probed the trypsin inhibitor activity of the purified fractions. And again, the process with dialysis steps shows to be more efficient in decrease the trypsin inhibitor activity compared with the process without dialysis steps, as we can see. But both levels can be considered good from the digestibility and of the fractions. I also proved the fluorescence measurements of the samples by the 3D emission excitation fluorescence matrices. And in this method, I used the samples in their dry state, so the powder. And this method measures all compounds that present some fluorescence properties that are present in our samples. In the case of PGMP flour, we got two different regions. So the first one corresponds to the region of emission of proteins, basically tyrosine, tryptophan. And the other region corresponds to the emission of unknown compounds that, according with the literature, can be a mix of different phenolic compounds. And an interesting thing is that with the desalting step of our purification process, we were able to remove the second region, as we can see here. So this analysis is a nice way to show the purity of our samples regarding these compounds. And other thing that we observed is that we had a shift in the maximum intensity peak that can suggest some change in the conformation of proteins from the partial hydrolyzed 7S globulin. I also probed the solubility of the samples, since it's very important from the functional properties. And I probed it in a different pH and also at low and high ionic strength. And as you can see at low salt concentration, we can observe a clear effect of pH on protein solubility with the classical U-shape of the solubility of proteins, when we have a higher solubility of the extremely alkaline and acid, and we have lower values around the isoelectric point of the proteins. And at high salt concentration, the native 7S globulin presented a solubility higher than 90% in almost all cases. And it's very nice. And other important thing is that the partial hydrolyzed globulins presented lower solubility compared with the native one. So this change in solubility, there are possible effects on protein functionality as well. So the main highlights of this part were that the 7S globulin was successfully separated using a protocol consisting of protein desaltings followed by anion exchange and size exclusion chromatographs. And the process without dialysis allowed the recovery of native 7S globulin, whereas the use of dialysis resulted in an unexpected partial hydrolysis of 7S globulin. And the partial hydrolysis decreased the solubility of PGMP 7S globulin. So in that way, at the end of this part, we select the native 7S globulin as a reference for functionality studies. And this leads us to the final part of the research that was focused in the air-water interface and foamy properties of crude and purified globulin extract. So here the idea was to investigate the foaming properties of PGMP globulins. That is a property very important for aerated products. They are foods that present foam-type structures. And currently in the industry, they're used mainly from agents of animal origin to produce these products. But there is a growing interest in studying possible substitutes of foamy agents of animal origin. And globulins stand up for being abundant in pulse-feed and also being soluble in diluted saline solutions. So in this part, the main question that we want to to answer was how geometry fractions performance from the agents compared to proteins of animal origin. So first, I investigated air-water interfacial properties of the protein suspensions using a drop tensiometer with the pendant drop method. In this method, a small drop of protein solution was formed in the tip of a needle in contact with air, and the drop was probed, was maintained with a surface area of 10 square millimeters, and we probed these properties at low and high protein concentration. And the idea here was to compare crude globulin extract with purified globulin. And from crude protein extract, I used commercial seeds and also seeds from Proline that was donated by Embrapa. And the native 7S globulin was used as a reference. So when we started measuring the tension, it started to decrease during the time of the experiment. And in the first time, we call it equilibrium period, where the drop is keep it in repose, and we can observe the migration of all surface agents that are present in the suspension to form the interfacial film. So we keep the drop at that way for 3 hours to allow the total migration of these proteins to form the interfacial film. And at this point, we can study the absorption behavior of the proteins by plotting the surface tension over time. So we have these results here at low and high protein concentration. And the main conclusion that we can get from it is that we observed a faster absorption of crude protein extract in both cases. They're represented by the green and orange lines. And this can be due to the presence of proteins of low molecular weight and also small non-proteinous compounds like phospholipids, for example, which consequently tend to migrate to the interface more quickly than the large proteins. In the second part, I applied some extension and compression circles to the drop that was formed. So here we apply the formations of 10, 20, and 30% in the surface area and in each amplitude step. So to study the surface dilatation rheology, we used 5 oscillatory cycles that were performed from each step. And I used the third middle cycles to calculate the storage and loss modulus, and also the loss tangent, that is the ratio between the storage and the loss modulus. And if you get a value higher than 1, we have a viscous behavior of our samples. And if this value is below 1, we have a predominance of the elastical behavior. So we're starting by the storage and loss modulus. Here we have the results at low and high protein concentration. And in both conditions, we observed a decrease in the storage modulus E when we increase the deformation amplitude. So this can be seen here and over here as well. So this means that the microstructure of the interfacial film was impacted by the high deformations that we applied, and also that we are outside the linear response region. Also, we can observe a higher decrease in the modulus E for the purified fractions compared with the crude protein extracts. And this means that the purified 7S globulin formed a more rigid layer compared with the crude protein extracts that presented less perturbation of the surface microstructure. And we should take a look in the loss tangent values. So in both cases, we obtained values below 1. So we have the predominance of the elastic behavior in our samples, but we observe some difference at low and high protein concentration. So at low protein concentration, all samples presented a very low loss tangent values, which means that our interfaces presented a highly elastic response to deformation. But when we increase the concentration, we start to see different behavior between the samples. So the purified 7S globulin maintained to show in the low loss tangent value, but the crude protein extract presented a higher loss tangent regardless of the applied deformation. So we can conclude some things about this. The first one is that when we have a loss tangent that is constant, the elastic contribution is independent of deformation, as we can see here. And when we have a higher loss tangent, that is the case of crude protein extract over the purified one, we have an interface that presents a lower tendency to relax to its initial organization, and also presents lower connectivity of the interfacial network. So to summarize all this, we have this schematic overview here. So at low protein concentrations, we observe a similar rheological behavior between purified 7S-globulin and crude protein extract. And this occurs probably because we have the dominance of the action of 7S-globulin in the interfacial film. But when we increase the protein concentration, we start to see difference between the samples. So here, the crude protein extracts, and probably with higher concentrations, other compounds begin to act at the interface, making it less rigid. So, and while the purified 7S-globulin showed a more rigid interface independently of the bulk concentration that was used. So the main conclusion that we can get from it is that the composition of the interfacial film is bulk concentration dependent. So to probe the foaming properties, I used a system of 2 syringes that were connected by a small tube. And in one syringe, I put 2 milliliters of protein solution, and in the other one, 4 milliliters of air. And the foam was produced by the back and forth movement of the syringe manually for 15 seconds. And after this, I kept it and I inverted the syringe in a support where images were captured every 1 minute. And that way, I determined the foamability, foam stability, and the volume of drained liquid using the ImageG software to process the raw images. So with this, the foam was distinguished from the empty space of the syringe by turning the threshold of the gray values of pixels. And here we have an example of a raw image and a treated image. So regarding foamability, I also use whey protein isolate, bovine serum albumin, and lysozyme as a control of foaming agent of animal orange. And I use 2 different types of suspensions, total protein suspensions and soluble protein suspensions only, that were suspensions after centrifugation. And we can see by the results that the type of the suspension was important from the crude protein extracts from PGMP globulin, and also for the lysozyme, which means that the aggregate state is more important in terms of foamability for these samples. And also I got a higher foamability from the 7S globulin that was purified from PGMP, the whey protein isolate, and the bovine serum albumin. And regarding the foam stability and the volume of drained liquids, we had a nice result that was a good performance of the 7S globulin that was purified. And it showed results similar to whey protein isolate. So it's a very nice result since we are compared with a protein of animal orange. And the other important thing is that the crude protein extracts presented a lower foam stability compared with the purified one, which can be explained by the interfacial properties results that I just showed. And the lysozyme presented the lowest foam stability, as we can see, and it was expected since we use it as a control of bad foaming agent. And regarding the drainage of volume during the experiment, we obtained a same range of volume that was drained for all samples. But if you take a look at the first minutes of the experiment, we will see that the lysozyme presented a faster drainage, while the DSA presented the lowest drainage of liquid. Regarding the physical appearance of foams, here we have a comparison of total protein suspension and soluble protein suspension. And we can see at the beginning of the process, right after the production of the foams, that we have more homogeneous foam systems from the samples when we use soluble protein suspensions only. This can be also seen from the foams after 120 minutes of storage, when we have more homogeneous foam system as well from the suspensions with only soluble protein. And the other important thing is that the samples from PGMP are very similar to the whey protein isolate, which is a very good result as well. So the main conclusions of this part involve that at low concentrations, purified globulins and crude protein extracts have a similar rheological behavior in the air-water interfacial film. But when we increase concentration, other compounds begin to act at the interfacial film and formed by crude protein extracts, making it less rigid. Also, the purified globulin show a better formability and foam stability than crude protein extracts and performance similar to a protein isolate and moving serum albumin. And finally, crude protein extracts from different seeds have different foaming properties, probably due to a different composition of non-protein compounds. So the main conclusions of this work were that about half of the soluble proteins in PGMP are 7S globulins, and the purification process that we developed recovered up to 30% of 7S globulins. And we observed a high level of mannose in globulin fractions, which can be an indication of glycosylation of these protein fractions. We also observed the partial hydrolysis during one of the processes that we use, which can affect the functionality of the proteins. And the composition of the air-water interfacial film stabilized by globulins from PGMP is low concentration dependent. And the PGMP 7S globulin has foaming properties comparable to whey protein isolate and bovine serum albumin. And we think in all these results together, we can separate it in this image here. So we started by a highly complex composition of protein fractions from PGMP. And so we went to a simpler system through protein purification. And finally, in the final stage, I studied the intrinsic properties of PGMP proteins that lead to new insights about the complexity composition of the protein fractions that we have. So some perspectives for future work involve to evaluate other protein fractionation methods, like the dry fractionation, and compare with the classical aqueous fractionation that we use here. Also to identify and quantify minor compounds, especially those that interfere with functionality of ingredients. And also to investigate other functional properties of PGMP protein fractions, such as emulsifying and gel forming properties. So this is the acknowledgement that I will do in the day of the defense. But thank you all there for attending, and I'm here for questions.
Thank you very much, Adilson.
You're welcome.
Thank you, Adilson.